Okay, here we are, Module 7. Continue our conversation on genetics. So last week we introduced the concept of the genes and that how our genes are actually carried on our chromosomes on the DNA, and that we represent these passing of traits from parent to offspring of these alleles. We get allele from both, one from each parent regarding a trait, and that often one allele or one trait will be dominant over another. So building on that, we're going to start talking about genetic disorders. So if an individual has what's known as an autosomal recessive disorder, well, right there, recessive tells you something. It actually tells you you have to be homozygous recessive in order to actually have the disorder. So let's look at this little chart here, and we notice that we have, okay, right here we have a parent, and we've got our female who actually is affected by this disorder because homozygous recessive. Married to a male, and he does not have the disorder. So we don't technically know if he is homozygous dominant. He could be AA, or he might be what's known as a carrier. He may be a lowercase a. We really don't know. All we know is that he is unaffected, right? But either one of these are going to be unaffected. So we're not sure which. So we're going to represent him by just A, because we know he's got at least one of these, and a question mark. Well, we're now going to follow this down, and we see they have two offspring, and both of them are unaffected. Now, we know because they are unaffected that their genotype is big A, little a. Now, how do I know that? Well, they're unaffected, which means they have to have gotten this from dad. Whether it was AA or AA, either way, a big A went to the offspring. Okay? Here and over here. They are unaffected. But we also know that the other allele has to be lowercase because that they would have had to have gotten lowercase from mom. So we know for a fact that these guys right here, actually these girls, they're female. Females are going to be represented. They're two daughters here. Females are going to be circles. Males are going to be squares in this example. Are both carriers. Right? Now the girls get married, the daughters, and once again they marry two unaffected males. They're unaffected. We don't know if they are homozygous dominant or if they're heterozygous. If once again, so we still have this scenario where we just put A with a question mark. Well, once again, we watch their offspring. Over here on the left, once again, we know this is an unaffected female. She's unaffected, so we know that she is A, little a, and then over here we have A, little A with a question mark, and A with a question mark. Now, how do we know, actually? How do we know all these people are unaffected? How do we know that these two actually have the little a's? Well, we look at their offspring. Okay, and this is the case where we have relatives mating. All right, they're actually cousins here. This is why first cousins shouldn't mate, but this could be anywhere in the animal or even plant kingdom for that matter. So anyways, this is why cousins shouldn't mate. We know because when you have these type of recessive disorders, if there's these relatives mating in here and having offspring, look what happens. You're more likely to actually pass on these homozygous recessive traits because their offspring are affected here and here. They're both affected. And then we have another unaffected one right here. But once again, we have no idea if it's big A, or big A, little a. All we know is that they do not show the disease. All right? So, let's look at this. Let's look at some notes here, straight from your book. Autosomal recessive disorders, which once again, the only way you're going to be affected is if you're homozygous recessive here. Affected children can have unaffected parents. Well, we saw that here. Affected children can have two unaffected parents. It's just that both parents are carriers. Heterozygotes have an unaffected phenotype. So once again, they are unaffected, but they are known as a carrier. Two affected parents will always have affected children, which makes sense. If I have AA mom and AA dad, the only thing that we can get here is allele, right? So two affected parents will always have affected children. Affected individuals with homozygous unaffected mates will have unaffected children. Well, let's think about that. That if I have an affected individual with a homozygous unaffected mate, that means we know what this mate is, they will always have what? Carriers for children, unaffected, but they're carriers. Close relatives who reproduce are more likely to have affected children. That's what this is meant to represent right here. Once again, two first cousins who reproduce, more likely to have affected children. That's why you're not supposed to marry your cousin. And both males and females are affected with equal frequency. Now, the reason that this is brought up is because in a little bit, we're going to talk about sex-linked traits. So they're traits that are actually on the XY chromosomes that actually determine your gender. And then all of a sudden, we're going to see it's very different as far as the populations of males versus females that carry on with that trait. Now let's look at an autosomal dominant disorder. So individuals that are homozygous dominant and heterozygous will have the disorder. So who's going to have the disorder? Well, once again, homozygous dominant and heterozygous. They're going to have the disorder. And so anytime you get this capital A in this case, they're going to be affected. The only way you're not going to have the disorder, so it's the opposite of the last one, is if you're homozygous recessive. So we have two affected parents up here that are big A, little a. They're heterozygous. How do I know they're heterozygous? Because they actually do have some offspring, some children that are unaffected. That's how I know. If they weren't both heterozygous, there's no chance that they'd get any of these. They also have two other children that are affected. This one, I have no idea if they're homozygous or heterozygous. This one I know is heterozygous. How do I know that? Because this person marries an unaffected homozygous recessive here, and of their offspring, they have one that is unaffected, which means this had to pass on, this little a had to pass on to this offspring. And then over on this side, of course, this unaffected homozygous recessive married another unaffected homozygous recessive, and there's no problems here. All right. So once again, going through the bullet points really quick, affected children will usually have affected parents. All right. Well, once again, that makes sense here. Heterozygous are affected, as opposed to before when it was only homozygous recessive. Two affected parents can produce an unaffected child. Well, how would that happen? Well, we saw that up here. Two affected parents, but they're both heterozygous, big A, little a, were able to actually have offspring that were unaffected. Two unaffected parents will not have affected children. Once again, we saw that over here. If you're unaffected in this case, you are guaranteed to be homozygous recessive, which means all your children will be unaffected as well. And once again, same thing here, both males and females are affected with equal frequency. It is not a sex-linked trait. A couple genetic disorders of interest that your book goes over, and there's a lot more than these. Tay-Sachs disease, lack of an enzyme that breaks down lipids in lysosomes, resulting in excess and eventually death of the baby. Obviously horrid here. But let's think about this. What are enzymes? These are those catalysts, which you know very well, that control the biochemical activity in a cell. Well, in this case, it's an enzyme that's going to break down lipids within those lysosomes, which are, once again, those little organelles that act almost kind of like little digesting, well, kind of like little stomachs within each cell to digest lipids and so forth. Well, if we're missing an enzyme, why would we be missing an enzyme? Well, enzymes are what? Most of the time when we talk about the word enzyme, 99.999% of the time, they're proteins coded on the DNA. So there is a genetic disorder, something's wrong with the DNA that codes for this enzyme. And so all of a sudden, it cannot make the proper enzyme to break out lipids. You have this excess in lipids, death of the baby. Cystic fibrosis. Probably you've heard of that one, a rather common one. In this case, chloride ions do not normally pass through cell membranes, resulting in thick mucus in lungs and other places, causing infections. Well, chloride ions pass through special channels, right? So imagine a cell membrane. This is my very poor picture of a channel here. All right. And chloride ions are supposed to be able to pass through. Well, what makes up these channels? Once again, it's, guess what? Proteins. And proteins are encoded on the DNA. So if there's a problem genetically with a protein that codes, the DNA that codes this protein for this channel, cystic fibrosis. Phenylketonuria, PKU. And once again, lack of a enzyme. Once again, it all has to go down. If we're talking about something genetically wrong, that means the DNA that's coding a protein, which is usually going to be structural, enzymatic, right? Something's going to go wrong there. So lack of an enzyme need to make certain, what? Amino acid, and affects nervous system development. You'll see little warning labels on like diet soda cans regarding people with PKU. Sickle cell disease. Red blood cells are sickle-shaped rather than biconcave that clog blood vessels. So a red blood cell, we're going to find out, kind of looks like a kind of a filled-in donut, if you will. All right, so what they call biconcave disc. Once again, a very poor drawing of a red blood cell. And they're filled with a special protein called hemoglobin, and they have to go through those blood vessels and through those capillaries, and they have to be able to squeeze through. All of a sudden, you get this weird sickle shape to it, and it doesn't actually flow as it should, and hence things can rather get clogged up. Lastly, Huntington's disease, really horrid disease that actually comes more into adulthood. And the Huntington protein has too many glutamine amino acids. So once again, it's a protein that's miscoded here, leading to what? Progressive degeneration of brain cells. So once again, a couple pictures out of the book. Here's a poor person with Huntington's disease on the left, and we can see the normal brain up here with all the different neurons up in here. But here, loss of neurons in a Huntington brain. Over here, looking at cystic fibrosis, here's the channel, here's that protein channel that's supposed to let the chloride ions go across, but it can't. So you get this thick mucus on one side of the lungs over here, and hence, once again, with cystic fibrosis, using a nebulizer and a special vest so they can breathe because of that thick mucus. Polygenic inheritance. So 2 or more sets of alleles govern one trait. All right, so think of it this way. We've been representing alleles by letters, right? Like big A, little a, or big R, big T, little t. So in this case, we're seeing 2 or more sets, okay, multiple alleles govern one trait. So each dominant allele, like big A, big R, big T, okay, codes for a product, so these effects are additive, which means a more of a variation of phenotypes. So it's a huge, more of a continuation. It's not round versus wrinkled like we talked about last week. This is where multiple genes are actually affecting a phenotype, affecting a different various trait. And this is what we see really most of the time, where we see hundreds or thousands of genes actually affecting the representation of a single trait, especially when we're talking about people. Environmental changes can also cause intervening phenotypes. We'll talk about that more in just a minute. Examples are skin colors, right? Can be very dark to very light, or height. Height varies obviously quite a bit. And that's because they are governed not just by, hey, tall or short, but there are multiple genes that have basically something to say about height. So that's polygenic traits. Poly meaning many, once again. Think about it, it's always in the words, many genic genes. So these are traits that are controlled by, oh yeah, many genes. Now what about multifactorial traits? Multi, another way of saying many, once again, factors, many factors that actually affect a trait. So this is a case where it's not just the genes that are going to affect the phenotype, but also the environment. Something from the outside, not just your DNA. And once again, our example could be skin color. Skin color is not just determined by your genes, but also by sun exposure. Or same thing, once again, we'll go back to height. Height can be affected by nutrition. If you're growing up and you are malnourished, you're not going to reach your full genetic height potential. So when we have polygenic inheritance and we also have these multifactorial traits, we're going to see, once again, more of a spectrum, not just on or off with genes, not just light and dark, but a spectrum from one extreme to the other. Whether we're talking about, once again, skin color, like up here, right? Skin color. And this is just showing two different genes being affecting it. There's actually more than that. But we also have height. Think about the wide range in height, and just this little example here, going from 5 foot to 6'5", and obviously you have a lot of people who are shorter than 5 foot and a lot of people who are taller than 6'5". But where are you going to see the most? In the middle of this bell curve here. Most people are going to be between 65 inches and 71 inches. So a couple examples of environment, affecting phenotype, that physical manifestation, once again, those multifactorial traits. We look at this Himalayan rabbit, and its actual coat, the color of its coat, is affected by temperature. Because there's an allele, so once again, think like big A, little a, there is an allele responsible for melanin production that appears to be active only at lower temperatures. So melanin, okay, special protein in skin that's going to actually affect the color of your skin. Well, the gene here is only active when it's lower. So the extremities that have lower temperature, like your ears up here, in this case, this Himalayan rabbit, the nose, the paws, they're darker in color because only those areas where the environment is having more effect on gene expression, if you will, that's where the rabbit is going to have the darker color because the melanin is active. So let me just get up here since the arrows are already up there. Incomplete dominance. Well, let's think about that. Occurs when the heterozygotes, so is intermediate between two homozygotes. So rather than, once again, something being completely dominant and versus recessive, we have multiple traits affecting. So we have affecting a trait. So we have kind of a blend, if you will. So we have this person over here who is curly hair, okay, and a whole different allele that says straight hair. And when they come together, they have this blend of wavy hair. So neither one is dominant. Neither one has complete dominance over the other. It's more of that blend, the in between the two. Versus codominance occurs when the alleles are equally expressed. Well, this is a little different. Okay, this is example, it comes from blood types, and you're going to learn a lot about this later on. But what happens is on our red blood cells, and I'm going to kind of draw another, here's my little red blood cell here. All right, so this is just kind of thinner middle area, like I said, a filled-in donut. There are these proteins around it, and we have a whole bunch of different types of proteins. Okay, and actually all cells, bacteria cells, all cells have these proteins, which we're going to call antigens. And antigens are what your body is going to actually look for with their antibodies. So those specialty proteins that are made to fight infection, to see what is foreign. So your body will make antibodies for self-defense, if you will, to fight off foreign invaders. How does it know? How does the antibody know it's a foreign invader? Because of these antigens on it, whether it be fungus or bacteria or what have you. So here's my little antibody. Okay, and the thing is that your body will not make, obviously, antibodies for your own cells, for your own, all right? So if we have type A blood, for example, type A blood, it means that your red blood cells have the antigen A on it, the A antigen. But what it also means is that your body, I'm going to make another one here. I'm going to have different antigens. We're going to call this, that's going to be type A. Another person who's type B might have, or will have, type B antigens on there. It's going to make these a little more circular here. So what happens is, if this is B, if I give someone with type B blood, and I give them, because of blood loss, I'm going to give them donated blood, type A blood, to them. Well, type B will have anti-A antigens. And the antibodies are going to latch on and attack that type A blood. Okay, so this is a little more physiology, blood physiology, than you probably wanted to know. But the point is, type A blood will have anti-B antibodies. Guess what? Type B blood will have anti-A. Type AB blood, well, type AB people have both antigens. So guess what? They don't have any antibodies for at least A or B, because their body sees that as self, not foreign. So these guys are often termed the universal recipient. And then if you're like me, I'm type O. So this is an AB person here. My blood cells don't have either antigen. So if I'm type O blood, I'm going to have both anti-A and anti-B antibodies. So I can't have any of these different types of blood put into my bloodstream, because my antibodies would go in there and knock them out. That being said, let's say I have a dad who is type A blood, and let's just say we know that he is homozygous here. Because let me point out to you, if their phenotype is type A, it could be A, big A, or it could be AO. Either way, because of this codominance, he's known as type A blood. He's going to have anti-B antibodies. Remember, type A is anti-B. Now I have on this side over here, let's say we have mom here, we have type B blood. And technically, we don't know if they're big B, or BO. We don't know. But then we have an offspring that is AB. So up here, Here's our offspring. Okay. The type AB blood has the characteristics of both blood types. Oh, well, let's look at my little pictures here. Here's type A with my little pointy triangles. Here's type B with my little rounded half circles. AB blood, which is represented like right here with our offspring, right? We have both our points and our little half circles, is what that's supposed to be. All right, so here we go. With codominance, AB is going to have the characteristics of both blood types, A and B. There's no dominance here that has both, codominance. Multiple allele inheritance. Kind of along these same lines, multiple alleles. The gene exists in several different allelic forms. That being said, okay, an example once again is this ABO. They could be A, they could be B, they could be O. Those are the three, whoops, ABC, ABO. Those are the three phenotypes. The different genotypes are represented by AA, AO in this case, BB, BO, or O. And lastly, don't forget AB. So there's actually all the different phenotypes. Here's all the different alleles. But a person only has a possibility of two. So here's one possibility. Here's another possibility, another possibility, another possibility, another possibility, another possibility. So once again, A and B are codominant. A and B are codominant. However, O itself is recessive. It's recessive to both A and B. So the only way you're going to be type O, which is, like I said, what I am, is to be OO. Should have represented that up here, OO. It's the only way you're going to be type O. So, based on what you know of what blood type, of what type of blood would be each of the following, okay? If we have a cross between AO and BO. So, once again, let's do a AO, BO. I know the answers are on the screen, but let's just draw it out anyways, all right? We have AB blood type, right? We have BO, which is type B. We have AO, which is type A. And we have type O. And once again, this is what we see. If we have a type A person who is heterozygous for type A, but A is dominant here, and we have a type B person who is heterozygous for type B, we could get, the offspring could be any of the four phenotypes. Type AB, type B, type A, or type O. So here it is again, another way of saying it. All right, for blood types, they have it as capital I here being the dominant, remember, and it's co-dominant, so there's a one that's represented by A, another one by B, lowercase I, meaning the recessive, the O, if you will. So we have type AB, type B, because it's a BO, if you will, type A, and recessive type O. And once again, what's the phenotypic ratio? A 1 to 1 to 1 to 1 ratio. You have equal probability in this scenario of getting type O and type AB and type A and type B. All right, that's it for this lecture. The next one we'll get into those sex-linked traits.